I'm Kevin Esvelt. I run a evolutionary and ecological engineering lab at MIT. We also spend a great deal of time thinking about how to advance biotechnology safely, which has led to some recent concerns given the events of the past year and a half. We are demonstrably vulnerable to accidental pandemics. We may not be sure exactly where SARS-CoV-2 came from, but it was definitely an accident. And in cybersecurity, people note that any system that is vulnerable to accidents is helpless against deliberate attack. And that's a little bit troubling because many thousands of individuals, including me, can build viruses from synthetic DNA. If you have a genome, you can order synthetic DNA, you can stitch it together using any number of protocols, and you can produce infectious virions. This can be done for most families of viruses today. And this is fine because we don't actually know which virus will cause the next pandemic, or even indeed which animal viruses could cause a pandemic in humans should they jump from the animal reservoir into a human. However, that's probably not going to remain true for long because many researchers are interested in figuring out which virus is likely to hit us next so we can begin preparing defenses, which would be lovely if the only way a pandemic could start was from nature. Of course, there is some associated accident risk of working with potential pandemic pathogens in the lab, but I'm really concerned about our ability to make these things from scratch now. Insofar as these projects, some of which actually aim to not only discover essentially all animal viruses out there, but rank them by the likelihood and severity of the pandemics that they could cause in humans, will eventually make it clear that there are credible future pandemic viruses that would take off in humans once introduced. In other words, even if you and I don't think that this is a good idea, and we perhaps ought to delay it as long as possible, we will eventually learn to make pandemics. If not through discovering natural viruses that could do it, we might learn how to enhance existing viruses to spread more effectively in humans. There's lots of plausible ways one can do this to make a fitness positive engineered element that will spread in the wild. CRISPR-based gene drive is one example. There will be more. And once we do that for a virus, thousands of people will gain access to these things. So right now we live in this world where someone discovers how to do this. Someone discovers a new pandemic virus, shows that it's credible. They try to warn the world. Of course, the message gets taken out in the current media environment. We know this is true. And we'll end up with somewhere in excess of 100, probably less than 1,000 well-resourced actors who can certainly build it, but we'll also have thousands, probably tens of thousands of individuals like me who just have the technical skills. And if we can get the commercial synthetic DNA, we can do it. We'd be much safer if we could limit access to hazardous DNA by screening all commercial DNA synthesis. If we had centralized screening for public hazards, then once there's a credible threat that's published, we can add it to the centralized database. And then what we might call the lone wolf actors wouldn't be able to get it anymore. The well-resourced folks still could because we assume that governments have enough people, or even folks who can throw tens of millions around can hire the relevant people to make their own synthetic DNA as needed. So they'll still be able to get it. That will be worse than the current world because the dotted line is the number of acknowledged nuclear powers today. And possibly an order of magnitude more nations could build nuclear weapons. But it would still be much better than it would be if the individuals can have access. But ideally, we wouldn't actually share information on how to build a future pandemic at all. We will stumble across it, but ideally the discoverer will notify the curators of the screening system. And even if the well-resourced actors find out slowly, particularly the nation states, then we won't actually have proliferation of the ability to ignite pandemics. So this is what we've been trying to do with a collaboration called Secure DNA that involves universities from all around the world. This is classic science diplomacy. And the basic idea is we had to come up with a new way of screening that didn't have false positives. So we essentially take a given hazard, break it up into chunks, pick fragments at random. We pre-compute functional equivalents, that is mutants that will still plausibly have the same function, screen all of those against GenBank to remove anything that would trigger a false alarm. And then we use a distributed oblivious pseudorandom function to essentially hash them all into gibberish. That's our database. Orders just go through the same process of hashing, which is a one-way mapping, so then we can just do a direct comparison. This, we think, will allow us to fully automate DNA synthesis screening using a single centralized database. And it means we can screen against things without the curator who adds it having to tell anyone else what it is. In other words, we can crowdsource risk finding and identify hazards, but those people who find them don't have to tell any other humans, other than perhaps one curator, what it is in order to guard the world against it. But even that is still going to leave a lot of people who could build pandemics as soon as we discover or learn to make pandemic viruses. So we need better defenses than last time. And I think one of the most obvious ones is we need to just sequence everything out there all the time. If we sequence wastewater, we know that we can find viruses that are present in humans and other pathogens as well, and quite efficiently too. And sequencing, of course, is increasing. The cost of sequencing decreases exponentially. So this will continue to improve with time. We also know if you sequence the river at the mouth of the river where it empties into the ocean, you can actually detect fragments of DNA from essentially all species in the entire watershed, including terrestrial ones. Meaning if we sequenced both natural waterways, marine environments, and wastewater, we could pick up anything increasing exponentially in the environment. And the key characteristic of anything that is a serious threat in biology is its exponential growth. If we just look for exponentially increasing Kmart fragments, then we'll know that there is something wrong. It might be a natural invasive species, it might be an engineered virus, but we will know that it's a problem, and it will not be possible for anyone to evade this kind of screening and detection system. Those are the two things that we're working on, but they're not going to be enough. So I'm putting this out there as a call. Many thousands of folks are going to be able to make pandemics sooner or later. What else should we be doing to prepare? Please let me know. Thanks.